Hello, everybody. This will be one of 3 animal labs. So for the next 3 weeks, we will be covering animals and learning how they evolved over time from animals with simple body plans to more advanced body plans. So this has its own module. This is Animal Lab 1 or week 14. Within the module, you'll see assignments. They will be worth 4 points out of your 12-point quiz grade. Please remember the quizzes are under the main menu of Canvas. There's a long list of menu options, including Confer Zoom, but in that same list are quizzes. Please make sure you go to the quizzes tab to do the next quiz, okay, which would be on the previous lab, not this one. But this lab, Lower Invertebrates, Animal Lab 1, those assignments are due the week after we cover this. Thank you. Just a reminder. Okay, now it's up. Okay, so here we're covering lower invertebrates. Here's an overview, though, of all animals. Animals can be divided into invertebrates or vertebrates. Invertebrates, if you feel like you're being surrounded by bugs and spiders, you are. They're more abundant than vertebrates, which we belong to. Okay. And so we're going to see in the next 3 weeks the evolution and development of bilateral symmetry. We have bilateral symmetry. In the next lecture, I'll explain bilateral symmetry versus radial symmetry and no symmetry, but not this week. Believe it or not, some animals did not have a head when they first arose. This process of obtaining a head is known as cephalization. Some organisms did not have a true digestive system, and some still don't if they're parasites. Others, we're going to see how the body cavity changes from having no body cavity to having a true body cavity, but we discussed that in another lab, as well as segmentation, repeating units. The first group of animals, which is the least advanced, are phylum Porifera, or the sponges. They did not give rise to other animals. They are asymmetrical. There's no way that you can see a mirror image of them. Some have radial symmetry. Okay, and maybe I should explain this now. So let me stop the share and go to a whiteboard to explain this. Now, I'm not the greatest artist. So you have different levels of symmetry. I'm going to do the ones you're more familiar with first, except I'm not good at drawing. So if we drew a butterfly, okay, there's its body, and there's its wings. I'm going to try my best here, because first of all, I'm not a good artist, and this is a butterfly. So let me fix it. Okay, so pretend the wings are equal size, but anyway, the butterfly is bilateral symmetry, has bilateral symmetry like us, meaning that you could draw a line in the center of the butterfly, like so, a vertical line, and you would have bilateral symmetry. meaning you can only draw a line one way to get a mirror image on the left and right. Okay, this butterfly is not perfect, but it still has bilateral symmetry. It has two wings on one side of my line and two wings on the other. Okay. Alternatively, you can have something with radial symmetry. I like to think of a round pizza. So hopefully you're not hungry. Okay, so here we have a circle. So if you buy a circular pizza, it has radial symmetry. Okay, and that means you can draw a line in many different directions and get mirror images. Of course, you can do a vertical line, just like we did for the butterfly, and get a mirror image left to right. Or you could do a line left to right and get a mirror image from top to bottom. You could even do a diagonal one or two. I'm not going to do a whole bunch because then it just gets crowded. You could do a diagonal line and then you'd have the same on the left side of the diagonal versus the right. So that's radial symmetry. You could keep going with the lines. Or you can have something asymmetrical. We'll see how I can draw that here. That's a little challenging. But you could have something like this. Okay. And it would be really hard to find a place, like if you did it here, it's not even, because if you did top to bottom, you have this little gap here that we don't have over here. If you did left to right, there's a big V-like shape, and then there's a semicircle here. Even if you did it diagonal, it would not be the same. So this would be asymmetrical, meaning there's no way to draw it so you have a mirror image. So this is asymmetrical or non-symmetrical. Okay, but we'll see some better pictures in a future lecture. Okay, so now let's go back to the PowerPoint. Okay, so just bear with me. Multitasking here. So again, sponges are usually asymmetrical, like shown on the last picture I drew. Some do have radial, but not as common. We'll see the next phylum is more likely to be radial. They're sessile, they don't move. Once they have gone beyond larval stage, they do not move. They're sessile. So they have to spread out when they're larvae. That's the only time they can take over the world, so to speak. They are only 2 cell layers thick, like the moss. They have an outer epidermis and the inner flagellated collar cells that help them swim. So they have these little whip-like projections that let them beat as one to help them with filter feeding. Now, filter feeding is a lazy type of eating, but if you can't move, that's the best way. They just keep their mouth open and the water current brings the food to them. It's lazier than drive-through. But interestingly enough, the first animals on Earth, like the sponges and the next group, they don't have a separate mouth and anus. So as they're bringing food in, it also goes out as waste. So they don't get all their nutrition. They have a skeleton composed of little specules or projections like body armor, sort of like a porcupine, made up of calcium carbonate. And we learned about algae, the coralline algae that had calcium carbonate. So that makes them be tough. And this is secreted by some amoeba-like cells. So here's a picture of sponges, a diagram on the left. They do have many pores, that's why it's called phylum Porifera, to help with water circulation, but its main mouth is the osculum that serves as the mouth and anus, and here's a picture of a sponge. Here we have phylum Cnidaria. The C is silent, just like the word knife, the K is silent. This includes a greater variety of organisms, unlike the sponges. It contains corals, hydra, sea anemones, and jellyfish. They have radial symmetry, as I drew earlier, like a pizza pie, pumpkin pie. I hope you're not hungry. They have some advantages over sponges. okay, in that radiosymmetry actually allows them to move if they want for food, but not all of them do. They have two body forms, an immobile polyp or sedentary polyp. Examples are hydra, or they can move like jellyfish do. Hope you can look at some virtual labs online. I think Aquarium of the Pacific has their exhibits online now. And they have a sac-like gut, which is very much like the sponges, meaning they only have one opening. You always hope your paper grocery bag only has one opening. You don't want to lose any groceries out the bottom. That's what that means. So their mouth and anus function together. So as you're eating, waste goes out. I hope that's not too gross for you. They have two cell layers like the sponges, but they have different names. One's called an epidermis and a gastrodermis. And they have a layer of jelly to separate them and help them with buoyancy or floating. Okay. Now they are more proactive defense than the sponges with the spicules. They can sting you. Hopefully no one's been stung by a jellyfish. They use that to capture their food, okay, and for defense, as I mentioned. Here's a picture of Hydra. It's bent over here with its tentacles looking for its prey. Okay, so Hydra. Now it's green, not because it's capable of photosynthesis, but it houses some algae to take in sunlight and CO2 to make sugars, as you may remember, for photosynthesis. We did that lab a while back. And so the Hydra gives an algae the safe place to live. And it can also give it CO2 gas from aerobic respiration that the algae can use for photosynthesis. The algae can share its sugar product with the Hydra as well as its oxygen gas. So they're in a symbiotic relationship which benefits both of them. Here's a picture of a jellyfish. Okay, this is of course the medusa form that moves. It has many tentacles. Okay, and it's also in phylum Cnidaria. You do need to know the phylum names. So I did talk about the Hydra or hydrozoans. They prefer to be the polyp form where they don't move, although as larvae they did move, just like the sponges. Jellyfish, which I showed you a picture of, the polyp form is reduced or absent. They prefer to move most of their life. And just trivia question there, they have horseshoe-shaped gonads. They make the egg and sperm. Corals and sea anemones, only the polyp form is present. Okay, you haven't really seen corals move. They do have stinging tentacles to capture their food, okay, but they don't move, so they have to grab it as it floats by. They have calcium carbonate like the red algae, coralline algae do, and that protects them from predation. No one wants to eat something that hard. Sea anemones do not have the calcium carbonate. They're the softer version of the coral, and they contain, like the Hydra, symbiotic algae. So here's some pictures of a medusa form. I'm sorry, it's misspelled. It's M-E-D-U-S-A on the left, and it doesn't move, but it has tentacles to grab food that floats by, or the medusa form that swims, that's on the right. Try to remember medusa starts with an M and mobile starts with an M. So here's a picture on the left of a coral, which are rock-like because of the calcium carbonate. And here we see a clownfish hanging out with the sea anemone, which does not have calcium carbonate. Then we go on to our first phylum of worms. There are a lot of worms in invertebrates. That's all they are. We have phylum Platyhelminthes. This phylum has worms that are free-living, which do not cause any problems, and others are parasites you want to stay away from. Platyhelminthes means flatworms. Think of the word plate is in there. They still, just like the sponges and jellyfish, have only one opening for digestion. So again, their waste goes out through their mouth. Advances, though, with Platyhelminthes versus the first two phylum are they have more specialized tissues. They have several well-developed organ systems, which they couldn't have before. But why do they have these? They have bilateral symmetry for the first time. So they have the same type of symmetry that I try to draw for the butterfly and ourselves and birds and most mammals. And most animals you're familiar with that are beyond this lab week have bilateral symmetry. This is the first time the animals have a head, a true head. This process of making a head is called cephalization. This having bilateral symmetry in a head allows localization of the sense organs, so they can more free to move safely. Here's a picture of various flatworms shown. The one on the left is my favorite. It actually looks like an arrow, and it is kind of cute for a worm. Number 3, you probably want to avoid. That's tapeworm. It will help you lose weight initially, but you'll get really sick from that. So this slide should say free living or parasitic. So some worms live on their own and don't cause any problems. Others are parasitic and will make you or a plant sick. The benign worm in this group is planaria. They feed on small animals or scavenge dead or organic matter. They do have little cilia for movement. In the protista chapter, talked about how paramecium moves with cilia. They used to have, as in Baji 120, in another building on campus, use fly swatters to try to catch flies to feed the planaria, but they didn't actually have that many flies in the classroom. Flukes are parasitic. They like to hang out in the liver, lungs, bladder, or blood vessels of vertebrates, which would include us. They have suckers to attach to the host internally. They can be monoecious, which you may have learned from the plant lab. That means that they'd have both male and female sex organs in the same animal, but they have mechanisms to make sure, just like the flowers did, that they don't self-fertilize themselves, because then there would not be any genetic diversity for evolution. Another word for monoecious is hermaphroditic. Now, there are some worms or flukes that are boys and girls, and that'd be dioecious. Us humans are also dioecious because we're both male or female when we're born. Primary host for an adult fluke is a vertebrate, so that means they prefer to live inside of vertebrates, animals with backbones. But while they're waiting for the right host, they'll hang out in a snail. Not all snails. These are mostly aquatic snails. So some people that go unprotected and go fishing by standing in the body of water are more prone to get these parasites than those that wear the boots all the way up to their hips, okay, or further. Tapeworms are nasty parasites as well. They're specialized parasites for living in the intestine. What are they enjoying? The food that you digested for them. They attach to the host by a scolex, which is a specialized structure with suckers and hooks. So they make sure they hang on to your intestine so it could grab some food. And again, they only have one opening. Following this head-like structure with the suckers and hooks, and you should see some pictures in our activities, are proglottids, which each of these segments has thousands of eggs per proglottid, and they leave through the infected organism's feces, and then the new host may by accident ingest them if they're near contaminated sites or near feces by accident. Also, we can get human tapeworm if the cattle or pigs that were ingested somehow ate worms that came from human feces, but that'd be very unsanitary conditions. And then if it's poorly cooked, it would cause problems with you. But you have to get the human version of the tapeworm to get sick. It's best to well cook your food. Initially, when microwaves came out to the public, people, I guess, believed the commercials too much and took raw food and put it directly in the microwave, and there were higher incidences of these parasites. Also, one of my students told me years ago, her husband got tapeworm and he was having incredible headaches. Fortunately, he had some good doctors that did some brain scans and found what the problem was, and he's okay. They removed the tapeworm from his brain. This is planaria, which is the benign flatworm. Okay, here it's looking at its nervous system. There's its head up here, nervous system, which includes the brain, so you can have localized sense organs. Now notice this worm has an ovary and testes, so it is hermaphroditic or monoecious, but there's mechanisms to make sure these sperm don't fertilize the same eggs in the same worm. Here's a picture of a liver fluke that you can get in unprotected waters if you're fishing, if the snails have the fluke. So these want to be the land snails. Here's a diagram, even though I described it already, of how you can get tapeworm. So this pig was exposed to human feces somehow, and somehow accidentally ate some with tapeworm in it. And then someone ate undercooked meat, and then they got the tapeworm. This enlarged structure, as what I described before, was the skolak, which has hooks and suckers to hang out in your intestine and eat your digested food. Because it is eating your pre-digested food, it doesn't need a digestive system. Instead, it uses yours, and then therefore all these little things after the skolaks can produce thousands of eggs, and now you've become a tapeworm making machine, unfortunately. Now your cat or dog can get tapeworm, but that's from ingesting contaminated fleas instead. But fortunately, they do have products to prevent fleas, so they don't swallow the wrong flea with the tapeworm. Then we have the last phylum for this week, phylum Nematoda. And we're going in order of advancement. So these are your more advanced species for this week. They're the most widespread of organisms, still worms. They have a complete digestive system. So this is the first time we have a complete digestive system, a separate mouth and anus. We start getting hints of a body cavity, but it's not a true body cavity. All the organs are there, but they're not protected by a lining, but there is some separation of organs. We'll go more over the different types of body cavity in another lab. They can also be parasitic as well. Whoops, sorry. Let me quickly go back to the end. Here's a picture here of some nematodes. Notice it says male and female. Would they be monoecious or dioecious? Mon means one, di means two. So these are dioecious because you have a separate male and female. Usually, most mammals are dioecious, and some worms can be as well, unlike planaria, which was both. But you don't memorize which one's monoecious or dioecious. But if we gave you a picture like this that said male and female, you should be able to say dioecious. Or if I gave you a picture like before with planaria, and you saw ovaries and testes, you would say monoecious. Okay. Here's a vinegar eel, which you won't get to see live. Please don't panic. It's not in the vinegar you buy in the store, the white vinegar for salad dressing or cleaning coffee pots. This would be if you didn't distill the vinegar. These vinegar eels like to hang out in vinegar, which is surprising because this pH is around 5. I don't know if you remember our discussion on pH and enzymes, but a lot of enzymes prefer a more neutral pH at 7. But these organisms are special. They like a slightly lower pH. This is trichinosis, which you can get trichinosis from. These are specialized parasites of pigs or pork, and they like to hang out in these little cysts in there. If you eat undercooked pork, you can get trichinosis or trichinosis, whereas any uncooked meat can be tapeworm, and flukes is usually from fish or fishing. This is a list of things you'd be looking at, but instead, of course, you're looking at the activities online. Okay, so let's go look at Canvas. So I'll stop this share and we'll go to Canvas, hopefully. Well, so we'll go to Canvas. Okay, so I'll be right back. I didn't share the screen with you yet. Share the screen. And here's our picture of Canvas. Okay, so you see this. I just want to remind everybody, because there was a little confusion earlier. Quizzes are here on the left, which is worth 8 out of 12 points. This week we are doing Animal Lab 1, which let's verify where it is. It should be week 14, I want to guess. I shouldn't guess though. So week 14, Diversity of Animals 1 is here. Lower Invertebrates PDF, that's what I'm going over or just went over right now. And I will post the lecture. And then you have some goals to read, as well as do read about sponges and do the activity on sponges. Phylum on Cnidaria, which includes jellyfish and hydra, coral and sea anemones. Then you have Platyhelminthes, which includes the free-living and parasitic organisms. And you have questions to answer. Phylum Nematoda. Okay, so these questions here go towards your 4 out of 12 points. Remember, I combine them together. The quiz under quizzes and this together. These questions are due a week after we do this lab, whereas the quiz you're doing would be the plant quiz this week. Okay, remember the quizzes are under quizzes. Also remember, if you have problems, please let me know as soon as possible. Thank you and have a good day. Bye-bye.